Okay. Let's start with carbs.
1 and 2.
Not really. But the video is going. But that's okay. What were we talking about?
Waffles.
Oh, waffles are good. I do like waffles.
I'm just so hungry right now, Jacob, that, you know.
Have you not had lab yet?
No, it's not till 3:15.
Oh, I'm sorry. Kind of. At least you don't have to run up a hill.
Don't get the butter ones. They make you feel awful.
Also, don't get gluten for it, then you can. Then you just have sugar water.
Oh, heck no.
Wait, what? Waffles are gross?
The butter, they give you 4 of those little like packets of butter you get like at the restaurant.
You have to eat them all?
It's like a
I
waffle
still
bar,
was
like,
like,
Oh, heck no.
Sugar water.
What? Disgusting.
The sugar water actually is
I just
I want
Just give me the protein.
When I was at restaurants, I always put the pink, like, artificial, like, sweetener or whatever in my drinks.
That's probably what it tastes like.
I don't know why. I always immediately regretted it after.
It's just your thing.
I was like, there's just sugar. And then I forget, oh, this is actually gross.
You forgot that it
I
was
forgot
gross.
that. I forgot that. I was like, I know I do this every time, so. there must
Welcome,
be a
Jackson.
reason.
Thank you.
I don't do that.
Oh, no. Oh, you walked in front of them?
Oh no, that's okay. You guys have been talking the entire time, so it's all in there.
You can just start it after you're done writing on it.
Yeah, I probably should have, but I don't really care.
Well, no, you can just.
It's true.
You still stop it.
It's not too much effort. Yeah. You could just end the video, start video.
I do that so many times. And what is it? And that's staying in the video. Okay, so carbs, you have the monosaccharides, glucose, galactose, and fructose. Fun fact, glucose and galactose, literally the only difference is, how many of you guys have taken OChem?
Yeah.
Yeah,
I have.
it's been a while.
Alright, cool. So this is very simplified glucose. Glucose has that. Galactose is that. That's literally the only difference. So that's why we can use the SGLT1 for both glucose and galactose, because they're basically the same thing. Cool. Okay, yeah, so monosaccharides. For us to actually get something useful out of them, we have to break them down into their monosaccharides. Oh, I should also put this in. Cellulose. Okay. They will be naturally found as disaccharides stuck together. 2 glucoses together is maltose. Glucose and galactose is lactose. Hopefully that makes sense because you've got the lactose and lactose. Cool. And then fructose and sucrose. You see the uck. Just make that connection. I don't know what you have to do. Sucrose is glucose and fructose. And then all of our polysaccharides, they're all chains of glucose. I don't know why it was made this way, that we don't use anything other than glucose for long polysaccharides. Amylose is literally just a straight chain of glucose. And so on. Amylopectin, so both of those are plants, and then glycogen is animal storage. So amylose is the simplified, that's your starch. So if you think of a potato, that's basically just all amylose. Amylopectin has some branches off of it. So you'd have like one branch here. Yeah, that's amazing.
Wow.
And then kind of so on. So amylopectin will have occasional branches. And then for animals and humans, because we're also animals, we make glycogen, which is basically amylopectin, but even more branched.
Is the storage and poly, whatever that means, are those like continuations of the monosaccharides, like glucose? Is that related with that, or are those just their own?
So all polysaccharides are made of just glucose. So it's just a bunch of glucose stuck
so
together.
all those are just glucose.
Yeah.
Oh, okay.
Just glucose. So glycogen, very, very branched. Amylopectin, kind of branched. Amylose, not branched at all. Cellulose is weird because it's also produced by plants, but the glucose molecules, it's like one's like this, and then the next one is flipped upside down, and then it's kind of like that repeating structure of them flipping every other one. So in terms of bonds that we care about, is always, so amylose is going to have an alpha 1,4 bond, amylopectin is going to have alpha 1,4 bond, and some alpha 1,6. I'll explain what those are in a second. Alpha 1,4 and alpha 1,6. And then cellulose is weird. It's a beta 1,4. And that's why we can't digest cellulose, is because we don't have enzymes that can break that beta 1,4 bond. So if I were to draw out what this would look like, oh, almost perfect. That's, nope. There we go. So your alpha 1,4 looks like that, very simplified. A beta 1,4 looks like this. Don't know why. All I do know why, but I don't care. Doesn't matter. That's organic chemistry. And then our alpha 1,6. Okay. We've got this oxygen and then like that. So that's an alpha 1,6. Branched, straight is your 1,4s. Yes, Tyler?
How important is it for us to know that?
I don't know, so I'm just telling it to you. He should have done that in class, but if he didn't, then it doesn't matter.
You did not.
Cool, it doesn't matter. I'll just tell you that because, like, in my mind, it helps me keep track of, like, all right, cool. So those ones I can break, this I can't, and then these ones require special things.
They're branched.
Okay, so I'm gonna draw something really scary, so don't get scared. Wow, I'm so good at this.
Oh.
Yep, nope, that got real bad real fast.
Is that a mouse?
It was an attempt. Okay, here's the head. Here's the nose.
What?
Yeah, I know. It's a physiology guy, not the drawing.
There's his teeth. Stomach.
Imagine that just like on the floor, just like.
You know those like chalk carvings that they do for like crime scenes? This is the chalk that you see. Okay. So for carbs, you have something called salivary amylase. Specifically salivary alpha amylase. It's specifically alpha because it breaks those alpha bonds, and this is going to only break alpha 1,4 bonds. But the issue is this only ends up doing like 10% of carb digestion. And it is inactivated by stomach acids. Remember, stomach releases hydrochloric acid, acid denatures proteins. So the salivary amylase is actually kind of useless because we eat something and then it just kind of immediately goes into our stomach and then the enzyme's done. So it's really, really dumb. Just know that that's a small intestine. S-I. Okay. Here's your pancreas. So what color do I want to use? What is red? Actually, we'll use green. Your pancreas releases something cool. It's another alpha amylase. But this one is pancreatic alpha amylase. And this one breaks alpha 1,4 bonds. So to give you an idea, the reason why I showed those bonds is because one of the enzymes that we have, it does actually matter. So if I'm breaking down something like amylopectin and glycogen, we have these chunks of alpha 1,6 bonds, these branches that alpha amylase cannot break down. So it can only get within like 3 or 4 glucose molecules, and then it gets stuck because this is stopping it. So we have to have some other enzymes that will help break that down. When we have that chunk that the amylase can't get to, we call that a dextrin. So, okay, any questions about this so far? No? Good, great. I'm gonna erase it. If you're worried, it'll be in the video, so don't worry about pictures. See, that's why I'm taking a video. Yeah, the video's been going for a while. Why? Oh my gosh!
I couldn't see the pancreas.
There's a video.
Right there.
Yeah, because on my channel I don't have any videos for Unit 5, so I decided I should probably make them to be nice to you guys. I like you guys better than last semester.
Oh, right.
Really?
Actually, yeah, I think I do.
We're gonna watch this video.
They should watch this video.
He said that to
Fuck
say
that.
and then he thought about it. There's a video going on, so don't.
Hi, Ashley. Welcome. Okay. This is the small intestine, just to orient you in your own body. Unless you're fasting. In that case, I'm sorry.
No, I can barely take notes right now.
Okay. So we're gonna have a whole bunch of disaccharides, monosaccharides, and then those things called dextrins. So we have, I'm gonna remove those. You know they're there. And then I'm gonna draw more cell over here that I hope is in the video. Okay, so I'm gonna try to color code this. So this enzyme is called alpha dextrinase. And it takes these dextrins and turns them into a bunch of glucose molecules. So it takes those chunks, is able to break that bond, and also breaks those alpha 1,4 bonds. So this one in specific is able to break those alpha 1,6 bonds, deal with that chunk of branched stuff. This one, that's a good blue marker, maltase. Hopefully you make the association maltase, maltose. This one is called lactase, which some people may not have. This one is called, oh, nope,
Fructase.
sucrase. Nice try. And so each of those are going to take their respective substrate, sucrose, turn it into glucose and fructose. Going to take lactose, turn it into galactose and glucose. And this is going to take our maltose and turn it into 2 glucoses. Good? Not that bad. Should make sense. Okay, so now we have a whole bunch of these glucoses and galactoses. And those are going to go through this beautiful transporter. Hopefully you remember how glucose and galactose are basically the exact same thing. So they can go through this same channel, which is SGLT1. Brings in sodium, glucose, or galactose. And then on this side, I have a GLUT2 transporter. I also, this one's really weird. This is GLUT5. Fructose is funky, and it thinks it's special, so it has its own dedicated transporter. And then all 3 of them are going to enter the blood through GLUT2. Good?
Mm-hmm.
Any questions,
Mm-hmm.
comments, concerns, declarations of random stuff?
Thank you.
Cool. All right. That's carbs.
Oh, 1 second. I'll take a picture.
It's in the video.
I'm not gonna
He's
watch it until
fasting.
later.
Yes, Ashley?
Videos.
He didn't talk about
Dextrin? So he talked about the alpha 1,6 bonds, and they're branched. So the alpha dextrinase is something that is able to break those branched points because amylase, I don't have it up here anymore, but amylase can't. Amylase can only break alpha 1,4 bonds. So once I'm left with that chunk of stuff that has those branches, I have to have something that breaks it down. Cellulose, beta 1,4, that's fiber. Plants, really useful. That's gives you good bowel movement. Okay, can I erase this now? All right. Someone says, wait, I don't feel bad.
Oh, I didn't take a picture.
It's in the video. Just watch the video.
I'll take a picture.
You can pause it and screenshot it.
Will the video have ads?
Will the video have ads? You think I get paid?
Jacob's actually monetizing that.
This video is sponsored by Raycon.
No, it'd be like this video sponsored by probably like NordVPN or something like that. Or Honey, I could do Honey.
Oh, that's a scam.
That's a scam.
Oh yeah, of course. What? Proteins. Proteins. We like proteins. Small intestine. Blood. And then I also need to have a little chunk here.
Really good.
That was a stomach sound.
And it was a stomach sound. Okay, so you eat protein. Pretty cool. Your stomach secretes something. Oh, that was fancy. So there's this little cell. Oh, which one is it? Is it the chief cell? I don't remember. We'll figure it out later. Releases the inactive pepsinogen. Pepsinogen is activated by stomach acid and turns into pepsin. Pepsin is a protease. It will start breaking down proteins into single amino acids. Pepsin is kind of weak sauce because it's pH sensitive. It's sensitive to very low pHs. So the moment that it makes its way into the small intestine, by carb release, it's inactivated because now I'm into more neutral pH. So pepsin doesn't do a ton of protein digestion, but it does start in the stomach. Yes, Tyler?
Does salivary amylase do anything to protein or fats?
No, that's only for carbs
So nothing gets remotely digested
in the mouth,
in the mouth?
except for carbs.
Okay.
Yeah. That's a lie.
Oh.
Sorry. No, you do have something called lingual lipase, which is a fat breakdown thing in your mouth, but we don't care. Okay, so now we've got a bunch of proteins. So, in response to partially digested foods, your pancreas, which I'm going to draw right here, pancreas, there we go. It's going to release a bunch of digestive enzymes, and those are, well, inactive ones. Trypsin, and I'm going to draw the other ones over here. So we have chemotrypsinogen, and then we have carboxypeptidase A and B. And that's all he wrote, and that's all we care about. A and B.
No need to know the endo or exo?
No, we're not going to worry about those. So the pancreas releases inactive enzymes. The reason they are inactive is because they are proteases. Proteases will just start chopping at anything that's a protein. It is non-specific. So if I release it active right here in the pancreatic duct, it will just start eating the pancreas. So I have to release it as an inactive enzyme. In the small intestine, I have this fun membrane-bound protein, which is called entero, meaning intestine, peptidase, splitting peptide, or also known as enterokinase. So what it does is it chops up some of that and turns it into trypsin, which is active. And then trypsin is going to go and activate all of those. because again, these are secreted as inactive, so it's going to turn chymotrypsinogen into chymotrypsin, and it's going to turn carboxypeptidase A and B into active carboxypeptidase A and B. And then all of these are going to start working at whatever protein you ate, start cleaving them off into single amino acids and then di and tripeptides. I'm going to, this is a small intestine, don't forget. Okay. So now I've got single amino acid, and I'm going to bring that into the cell along with sodium. I didn't write it, but don't forget this, but you have the sodium potassium pump on every single cell. Remember, it is the most important cell forever and always and for all time. So it's running here, gradient goes. But these di and tripeptides, which I'm going to write like that because I'm lazy, di and tripeptides aren't single amino acids because our body only uses single amino acids in the blood. So we have to have some way to get these broken down further because those enzymes, these are too small for them to work on. So I bring in these di and tripeptides with a hydrogen antiporter, and then I've got intracellular proteases. Think like lysosomes and stuff from your Bio 180 days. And then it's going to take those and turn them into single amino acids. And then all of those are going to be dumped into the blood. And I think it was with the sodium antiporter, but I'm not entirely sure, so you may have to double check me, because in the book I've seen it as this, but I've also seen it with the sodium, so I guess it could be either or. That's proteins.
He wrote it with sodium.
You wrote with sodium? All right, cool. Then we're going to go with that. Sodium comes in, pushes the amino acids out. And then you just get a bunch, what, other way around? Both go out? That doesn't make sense.
I think.
You did draw it.
That's disgusting. Well, all right.
I asked him about it in his
And
office and he said he comes in.
why did you say something earlier, Ashley?
I gotta look at my notes now.
Sodium
It
comes in.
didn't make sense.
Sodium goes in because it goes down its gradient, pushes these out. If anything, I'm pretty sure it should actually just be amino acids going out because I'm getting a bunch in here, just down the gradient. So in the textbook, that isn't even there. Fun fact. So we have it all on video, so there are many different possible options. This is great. Okay, any questions about proteins? This is it.
What were you saying about the entero and
So-
protease and kinase?
Same name for the same enzyme. This enzyme is bound membrane on small intestinal cells, and it's what turns trypsinogen into trypsin, making it active. Because we don't want it to be active until it's in the small intestine.
There's something like M cells.
We'll talk about that. Actually, I guess we could do that now. So M cells, I'm going to erase the stomach. You know what happens there. Food. Why would you say that? Okay, so you can draw like that. That looks amazing. So if you were a baby or very, very young, you have small gaps between your intestinal cells, and larger chains of proteins, which I will just draw like this, can sneak their way in. And there's this space, interstitial space, before stuff gets to the blood. And this is where your white blood cells sit, kind of like just ready for any kind of potential infection. So when these proteins come in, they're going to come in and sample it and make sure that it's not a pathogen and trying to kill you. M cells, because as you grow up, this junction is no longer there. So the M cell is going to pull in peptide endocytose, and then push it back out. So now I've got this peptide out here that the white blood cell can come along, check out, and be like, is this good, bad? And if it's bad, you get an allergy, and if it's good, then nothing bad happens. Good? That's your M cell. It's just kind of like helping the immune system determine if there is anything unsafe in the food that you ate. Good? You ready for lipids? Lipids is a lot. If you want a picture, take it now. Yeah, I know. I knew there was at least someone. Okay. Even though I talked about lingual lipase, we don't care. It exists, but so what? Okay. I hope you guys have come to appreciate your pancreas. Your pancreas dumps like literally everything into your small intestine for digestion. So if you don't have a pancreas, pancreatic cancer, no bueno. You can't eat anything. I mean, you can, but you don't get anything out of it, and it's really sad. Okay, so we're gonna draw fats like, you were so close to greatness. Maybe this one will work better. Oh, much better. I don't know why I draw them like this, but I do. So those are triglycerides. That makes up a large portion of the fat that we eat normally. Because they are hydrophobic, they basically form like a little ring around each other called a micelle. So your micelle would be like this. All the hydrophilic parts on the outside because they're fatty acids, and then all your carbon tails pointing on the inside so they doesn't have to interact with water as much. So the issue is this is kind of dumb because you can't digest this. It's too big, and the lipase that is released by the pancreas can't work on this. So what happens is at the same time, we have two different things. Kind of like that little nub right there. So there's your liver, there's your gallbladder. The liver and gallbladder release something called bile salts. So bile salts help emulsify fats. So instead of being like this, I'm just going to draw these kind of like this. They help break it down into smaller, slightly more manageable chunks. The issue is bile salts inhibit the activity of the lipase that the pancreas produces. So the pancreas also produces something called colipase, which I'm going to draw in green. So green is our colipase. Blue is our bile salts. And then that allows lipase to come in here and start breaking down these triglycerides into monoglycerides and free fatty acids. So lipase, that's what that says, is lipase. So it turns it into monoglycerides and free fatty acids. Because these are still nonpolar hydrophobic, they can pass through the membrane really easily, and then through lots of fun packaging in the Golgi apparatus and Smooth ER, they are reformed, which is kind of dumb, into triglycerides and packaged into a great big chunky thing. They also have some other stuff which I'm going to use. Let's use red. That red is cholesterol. And this big chunky thing is called a chylomicron. It's funny that I call it big even though micron is small. But that's okay. This is then exocytosed, but it's too big for the blood. So thankfully, yeah. So my chylomicron is pushed out into my interstitial space and then sucked up by the lymph vessel. Those will end up going into your subclavian veins and then dumping it back into your blood. And then it's going to start circulating around there. And then, now we'll move on to lipid metabolism. Any questions about this? This is just the digestion of fats.
Can you say again, like, once our monoglycerides are ever across the membrane, they just form up with a chylomicron type thing?
Yeah, so
Oh, right, through the Golgi. Okay.
yeah, so it's kind of weird. The Golgi apparatus and smooth ER and rough ER, I don't know which ones. The ER in general is going to reform your triglycerides and then package them into a chylomicron. And you're like, that's kind of stupid because I basically had one right here anyway. So, Ashley.
So cholesterol, isn't that fat by LDLs?
We're not there yet.
But how does that?
So your cells will make cholesterol, and your, like, all of your tissue needs cholesterol because it's part of what makes your plasma membrane semi-fluid. So any cholesterol that I do have in here that's like extra, I will chuck it into the chylomicron to deliver to the rest of the body. Braxton.
Are those just proteins that it's in or something? The chylomicron? Is that a protein?
That stuff? Or the red?
Is the green thing the
Yeah, the whole thing is called the chylomicron. There's also
like a lipoprotein?
yeah, so this is called a lipoprotein. This thing is called ApoA I. That's part of what makes it a protein. So this protein is what holds the chylomicron together. You will need to know this because there are receptors on tissue cells that recognize that ApoC2 protein. Tyler.
You said exocytosis is where? Or what's that? Just
Takes the chylomicron, pushes it into the interstitial space, and then it gets by the lymph.
That's all.
Yep. That's the digestion part. Are you ready for the big, scary, massive stuff?
No.
Okay, any other questions before I start to go off?
Oh, go on.
Ashley?
Isn't it the ApoE that it has the receptors for?
There are a lot of receptors for everything. So if I end up saying something wrong, you will have to correct me and be like, Jacob, I don't know about that. But look it up in the textbook first before you try to correct me. Because I don't know if I'm wrong. Tyler?
The picture that he drew, he had something with FABP. Do you know what that is?
That.
Oh, that's the fatty acid binding protein. So because these are lipid soluble, they don't like water, when they get back in here, they start forming a micelle again. So the fatty acid binding protein attaches them and then brings it to the Golgi body and the ER. Good? All right, cool. Time to scare you guys. Actually, I'm going to try to avoid doing that. Too late.
Just kidding.
Okay. So here's our chylomicron. And it's going to flow around in the blood. I'm not going to draw the blood because I'm lazy. But on it, it ends up picking up this thing, which is an ApoB 48. And then which one did I draw? I think it was purple. And then it has ApoC2. And then if I remember correctly, it does have ApoE, but I don't think we care. I don't remember it ever doing anything.
Yeah, on the drawing Hunt did, it was ApoE and ApoC2.
That's the
It
one.
does have ApoB48,
Oh,
but we don't really care. That makes it water soluble.
We don't care about the ApoA 48.
And that's why I wrote it in brown and you can't really see it. So it's like barely showing up in the video, so I mean that's fine. Okay. So this is gonna go around in the blood. So this is always in the blood. So I'm not gonna draw the blood, just understand that this is happening in the blood. So as the chylomicron goes around, it's going to interact with some kind of tissue cell. The tissue cell has this fun enzyme. Ooh, what is it called?
Lipoprotein lipase.
LPL, lipoprotein lipase. Thank you, Braxton. So LPL is going to recognize. All of you guys are ending up featured in this video. If you talked at all, you're in here.
I need to be monetized.
This video is sponsored by NordVPN.
Video sponsored by BYUI.
Go Cougs. Or,
Go, no.
Yes,
Vikings.
Yes, Vikings.
Or Vikings? That's
Yeah, go Vikings.
Yeah, go
Yeah.
Vikings, baby.
Back in Rick's.
I thought we were just like an eye or something.
It's an eye from
The
Pixar.
island.
I don't even know who they are.
Okay, so the tissue cell has that ApoC2 receptor. It recognizes the ApoC2 on the chylomicron, and the chylomicron dumps triglycerides into the cell because our cells use fats to turn into energy. It's called beta oxidation. It's a really fun thing. Biochem. Anyway, so that happens. As the chylomicron continues to do that, it loses more and more of its stuff and becomes a chylomicron remnant. The chylomicron remnant is then going to go to this really cool organ called your liver. That is actually such a nice liver. Yeah, I'm pretty proud of that. It's not going to happen again, just so you know.
Square.
I would never draw a square.
Square.
The chylomicron remnant is then eaten by the liver, and then we have no more chylomicrons. Good so far. Did he talk about what specifically the liver recognized on it?
ApoE.
The ApoE?
Yeah.
All right, fine. I will draw the
Yeah,
ApoE.
you wrote that down.
Recognizes ApoE eats remnant. The liver is basically your ginormous storage, eat everything organ. It's kind of crazy. This was the good orange. That's my bad orange. Okay, then the liver is going to produce something called a VLDL. We're going to draw this. We'll keep using green. So the VLDL produced by liver, this has cholesterol, triglycerides, something called cholesterol esters. Just know that it's got a bunch of fat stuff inside it. And it ends up picking up our, where's the blue? It gets ApoE. And if I remember correctly, it also gets ApoC2. And then, because I don't care, I'm gonna write it here. Gets ApoB 100. Really weird. So just kind of like emphasizing the difference, ApoB 100 versus the ApoB 48. Both of them are used to help it be soluble within the blood because blood is water, these are fats. So it's just a different protein to let it through the water.
You know hepatic lipase?
We're not there yet.
Oh,
Soon, Braxton. You're jumping the gun.
He's just trying to be on the video.
He's gonna plug his own Instagram.
Okay. So again, tissue cell has that ApoC2 receptor, and then VLDL dumps triglycerides, and then it becomes what we call an IDL. With the different names, this stands for very low density lipoprotein. This stands for intermediate density lipoprotein, because as they dump more of triglycerides, their other stuff becomes more concentrated, so we call it more dense. It's kind of stupid, but it is how it is. Okay, so this still has ApoA I, and that's really the only one that we care about. So this is going to be super confusing, so I'm just going to draw another liver. Yeah, still not too bad. Not as good as that one, but what?
What might be better?
Oh, I appreciate it. Okay, so this is going to do one of two things. And it's a 50% chance for it to go either way. So the liver can either recognize the ApoE and just eat the whole IDL. On the other hand, and we'll draw this in purple, this is our hepatic lipase. And hepatic lipase recognizes the ApoE as well, but instead of just pulling in the whole IDL, it takes more triglycerides, but leaves the rest of it out. So what happens is it goes through the hepatic lipase and then becomes our LDL. LDLs, super duper concentrated. If I remember correctly, basically all that they have left is that ApoB 100. So it's basically just kind of like this floating ball of fat all around in our body. Not very helpful. The nice thing is there are some cells in our body, some cell, and it's got this little receptor that's called an LDL receptor. What it does is it recognizes that LDL and just eats the whole thing and then breaks it down, uses it for cholesterol and the fatty acids that it's got left. And then the LDL is gone. Any questions? Jackson?
Do the IDLs go back to the tissue cell as well? Or just to the liver?
They can. So they can go back to it and eventually lose their ApoC2. But I don't draw that because we don't care.
Okay.
Then they had something with epithelial cells. It's like going to like the VLDL. It was using ApoA I. It was saying it takes fat to liver. Maybe it's not important. I don't know. He's using HDL.
Oh yeah. That's where I was about to go. Again, you just keep jumping.
Sorry, I just trying to complete my picture.
You're fine. Here's your small intestine, and it releases this fun little protein called ApoA I. ApoA I is going to float around in here in the small intestine, and it's going to start picking up free fatty acids. So I'm gonna use, not because that's the gross orange one. Just gonna start picking up free fatty acids, forming around it. And it's gonna slowly start to like form a little ball. And eventually it's gonna make its way into the liver, where the liver is going to package it as a full-on HDL, which I'm going to draw as this little blue ball. I don't know how to explain it, so I'll just say it again. ApoA picks up your free fatty acids from your blood and inside the small intestine, starts to try to form kind of like its own little packaged unit thing. It's going to float through the blood. The liver is going to pick it up, and the liver is going to fully package it as an HDL, so high density lipoprotein, because it's going to have a lot of protein, not a ton of fat, not a ton of cholesterol. So then, this is really interesting, and I don't know how to explain it based off of my drawing, but the HDL and VLDL will kind of like meet up in the blood, and they'll swap some stuff. So the VLDL will dump off some triglycerides, and then, just kidding, the VLDL will dump some cholesterol into the HDL, and the HDL will dump some triglycerides into the VLDL. Did that make sense? I'll draw it.
So
VLDL. So, lose triglycerides. Gains cholesterol. That makes sense. Wait, that's wrong.
Yeah, we're all like, yeah, makes sense.
So the VLDL is the one that gains the triglycerides. The HDL is the one that gains cholesterol. So loses. Now that should make sense. What?
So it just seems like if you're just swapping it and you don't include loses.
Okay. As long as you understand it, we're good. That's lipids. And then HDLs will float around and eventually get eaten by the liver. And then it will turn it into bile salts, and then you poop it out.
I'm so sorry. Can you start from the beginning and just run through it?
Run through it real fast?
Yeah.
Okay. So, digestion, chylomicron. The chylomicron has these receptors on it. Fun fact, it actually ends up picking them up when it interacts with the HDL. How? I don't know, but it happens. So it's got these receptors on it. It's going to flow through the blood, and it's going to occasionally bump into cells that need fats. And so that lipoprotein lipase enzyme on the outside recognizes the ApoC2 protein on the chylomicron, and the chylomicron dumps triglycerides into the cell. As it keeps doing that, it loses more and more of triglycerides, becoming smaller and smaller, and becomes a chylomicron remnant. The chylomicron remnant is eaten by the liver, and then the liver processes all those triglycerides and fats and turns it into a VLDL. The VLDL has the same ApoC2 receptor that the chylomicron had, so it's going to do the same thing. It's going to go to tissue cells, lipoprotein lipase, detect it, sucks in fatty acids. As that happens, it becomes smaller, turns into an IDL. IDLs floating around in the blood will do one of two things. So it can either...
Sorry. So you said VLDL attaches to the tissue and then turns into an IDL?
Yes, when it dumps triglycerides.
Okay.
So now we've got a smaller thing, and then the IDL is going to interact with your liver. The liver will either just eat it and then break it down, or it can go through hepatic lipase. Someone may want to check me on this, but I'm like 75% confident. So it's going to go through the hepatic lipase, where it's going to pull out even more of those fats and triglycerides and then turn it into an LDL. Or the liver will just take it up, eat it, and then turn it into other stuff. Those LDLs will float around in the blood. They're high in cholesterol, not super high in fat. And that's kind of where we get that atherosclerosis stuff that Hunt was talking about beforehand. Your white blood cells can eat this, and then they turn into what's called a foam cell. And that's where you get that buildup of plaque on the edges of your arteries. HDLs are really nice because they'll come along and they'll pick up cholesterol from these as well. Some tissue cells have LDL receptors, and they'll take in that LDL, break it down, and then they'll use the cholesterol and free fatty acids that are in there. At the same time, your small intestine also produces a protein called ApoA I. ApoA I starts to attract free fatty acids around it and starts to form kind of like this circular, almost micelle-like structure. And then as it floats around in the blood, the liver picks it up and packages it into an HDL, making it completely spherical and very nicely packaged. HDLs will float along in the blood. They'll pick up cholesterol from LDLs, and they'll also interact with VLDLs, give them their protein receptor things, and also give them triglycerides, and then pick up cholesterol from the VLDLs as well. And that's lipids. Okay, Jackson first and then Ashley.
You mentioned something about LCAT was being raised due to HDLs?
LCAT.
Or is it ApoA or something?
No, I have that.
Go into the picture.
I'm trying to remember what LCAT is. So that's probably the enzyme that recognizes the HDL. That's because the HDLs will float around in the blood and eventually get metabolized by the liver. And that LCAT enzyme is probably, oh, no, that's what it is. Okay, I remember. So the LCAT is what makes this almost micelle thing into the HDL. That's all it is.
Oh, okay. So it doesn't go through the liver?
Kinda, sorta ish.
Alright.
We don't care. We just know that the LCAT is what turns this into that. Here, I'll write that down.
So when you're increasing arrow on LCAT, it's just meaning there's more HDLs
Mm-hmm.
being produced.
Turns this into that. All right, Ashley, you had a question?
So HDLs gain cholesterol and VLDLs gain triglycerides.
Yes. All right. Any other questions? Perfect, you guys got this? Okay. All right. Video done.